of the slide. Okay, so now we know already there are 5 kingdoms. So the first kingdom we'll be starting today, that is kingdom Monera. Now, the most important thing that you need to remember here is actually that, like I had said earlier, the progression of the classification has been decided in such a way that from lowest complexity to the highest complexity of the organism, they are segregated into different categories. So the first one that naturally comes being unicellular and then being prokaryotic. So unicellular, we know, unicellular, these are single-celled organisms. And then they are also prokaryotic in nature. That means these are without any nuclear development, without nuclear membrane and so on. So that's the prokaryotic. Now, yes.
Sir, but if an organism is made up of a prokaryotic cell, isn't it necessary for them to be unicellular? Because prokaryotics are not multicellular.
Correct. So prokaryotic does not mean they have many cells, or even what you see. For example, there is one example here which is given. There are multiple cells, but this is not one organism. These are many organisms or many bacterial cells living together. So they are always unicellular. Kingdom Monera, all of them, they will be always unicellular.
And so we can just write they're prokaryotic, right? Because then it automatically becomes unicellular.
Correct. But then prokaryotic, oh, you mean it that way? Yes, that is true. That is right. But then there are group of organisms, they stay together. And they look like multicellular, look like I'm saying, they are not multicellular at all. Because they are just different. Like if we say within a family, we say 5 or 4 individuals, that does not mean we are one, right? We are just together somehow. So this is also something like that. Some of the bacteria, they have a tendency to stay together with each other. And that's when they look like multicellular, but they are not. They are still individually unicellular prokaryote.
Okay.
Okay, so then these kingdom Monera that mainly will include bacteria, the blue-green bacteria. Now this is a special type of bacteria which is able to do photosynthesis, and some unicellular algae. We will see algae what they are and how do they look like in maybe the next part, next kingdom. But the main component of this kingdom Monera is bacteria. And you all know what bacteria are probably, because you would have studied the prokaryotic cell or bacterial cell. So they have no organized nucleus in a single cell. The nuclear material, that is DNA, is distributed in a cell without any nuclear membrane. And that's why the condition is prokaryotic, which is quite obvious that we know. And of course, because they are prokaryotic, they don't even have these organelles like mitochondria or chloroplast. Right? In some of the autotrophic bacteria, chloroplast may not be present, but chlorophyll-like pigments, they are present. The whole structure of chloroplast is more of a eukaryotic in nature. But what is most important within the chloroplast is that protein complex system of the chlorophyll and so on, right? So if that is present, that is good enough for synthesis or for the photosynthesis process. So that's when these prokaryotic organisms, they lack these mitochondria and chloroplasts. Okay, let me go to the next one and then I'll come back to that picture. So in general, the bacteria, they are present everywhere. So like if you take water bodies, if you take plant, tree, animals, anywhere on the soil, in the atmosphere, everywhere bacteria, they are present. And they are microscopic in nature. You can name any space around us, not the above atmosphere. you will always find bacteria in that particular space. You can imagine the amount of bacteria that we will have in this like whole of the.
There might be millions of bacteria on my pen right now.
Yeah, exactly. So you can imagine the total number which is present all around us. Except there are these research laboratory or pathology laboratories or some experimental setups. They do not have this bacteria because they are kept sterile for the experimental work, which is quite obvious. So that is, these are the only places, but they are made artificially bacteria-less without any bacterial presence. Otherwise, each of the space around us will always be with many bacteria. They possess cell wall and are prokaryotic. They can be heterotrophic or autotrophic. Now again, heterotrophic means they are dependent for nutrition on the other organism. That is what is heterotrophic. And they are autotrophic. That means they are able to synthesize their own food. So they can be of any of these type. Most of the bacteria, they are heterotrophic. There is a small percentage which are autotrophic. That means because they are and these are like, I would say, they are aquatic bacteria, because they are present in the water bodies, which are most primitive form of this bacteria. The major difference that you would remember from prokaryote and eukaryote, that the cell wall of these bacteria or kingdom Monera organisms, it is formed of amino acid and polysaccharides, which ultimately means proteins and carbohydrates, that is polysaccharides particularly. And these heterotrophic bacteria, they can again be parasitic and saprophytic. Does anyone know what is saprophytic? Saprophytic? That is heterotrophic. Anyone from the other side? No. Parasitic, I assume everyone knows that they grow on the other living organisms. That is parasitic, so that they can take or suck the nutrition from the host body or host cell.
So then that will mean that the saprophytic will not need a host cell and they can live on their own. Just a guess.
In a way, right? So saprophytic are those bacteria which lives on dead or decaying organisms. So parasitic will live on the survive on the living organisms. Saprophytic will survive on the dead or decaying organism. Yajnesh?
So like mushrooms are saprophytic, right?
Correct. Mushrooms, but not all the mushrooms. Some of them, they are saprophytic. Yeah. I hope everyone understood the saprophytic part. Right? So they are able to survive on the dead or decaying. Decaying means as if like something has been dead and then it degrades over time. That is what we call as decaying organism. So, and again, in case of autotrophic, so heterotrophic can be parasitic or saprophytic, and autotrophic can be chemosynthetic or they can be photosynthetic. That means they can either use the chemical energy, which is in the form of ATP or some other bond breaking molecule, or it can also be in the form of photosynthetic, that is the light energy, and then they can synthesize their own food. That is what is autotrophic. So either using chemical energy and then doing function of photosynthesis, that is chemosynthetic autotrophic. And then photosynthetic autotrophics are the using light energy, they perform photosynthesis and synthesize their own food. That is photosynthetic autotrophic. Any question until here? Anyone? These are just few characteristics here that you need to remember. Most important ones being they are unicellular and prokaryotic, and then you can write all the characteristics of the prokaryotes, right? They have no nucleus, no a single cell wall, and all those things. No cell organelles, and most importantly, the nutrition. Okay, so then there are several types of these bacteria, which are classified in general into 3 or 4 categories. There are many more types, but major types are these ones. What we commonly called as coccus or cocci. These are the bacteria in spherical shape. So all these types, they are based on the shape of the bacteria. So for example, if you see in this diagram, you can see there are round, elongated, spiral, and then there is something like this, which is comma shaped, we call it, right? So these are the main types. Otherwise, all of them, these are the clustered spherical shape. These ones are elongated rods attached one after another. These are spiral shape. These ones are also spiral shape. So that way, the shape will remain common to most of the bacteria. Just their arrangement might differ. So based on that shape, they can be cocci, which means spherical, bacillus, which means rod shape, and coccus and cocci. Coccus is the singular and cocci is the plural. Same way, bacillus, bacilli, vibrium, vibrio. That is the comma-shaped bacteria. And spirillum, this is the spirilla, which is the plural form, and they are spiral shaped. So these are the 4 main types. Now, in terms of examples, like if you see here, again, within cocci, there will be many types. Monococci, diplococci, triplococci, streptococci, staphylococci. There are several subtypes. Something like this, you can see staphylococci. They are having this grape-like arrangement. That is staphylococci. Diplococci, that means they are present in 2 cocci. That is 2 spherical. Again, this you need not remember. This is just for your information, but you should also know that there are different types based on their arrangement. Again, a tetracocci, and then there is something called as sarsinia, right? That is a cubical arrangement of the this cocci bacteria, and that way there are these cocci or the spear-shaped bacteria. Then there are rod cells. Again, same here. If it is monobacilli, dibacilli, streptobacilli, and so on. So there are several such types. They can have either this flagella around them, or they can have this spore-forming structure on the side. So that way, again, there are several subtypes, but again, you need not know it. What you need to remember is spear-shaped cocci bacteria, bacilli, these are rod-shaped and so on. And one examples of it. And the spiral-shaped bacteria, these are literally spiral-shaped. For example, this Vibrio, which is categorized separately in some reference books, because it's like more like a comma shape. So this is like the round part of comma, and this is like a tail of that comma. So that way, this Vibrio, that is one organism. You might know the disease cholera, right? So Vibrio cholerae is the organism which causes that disease, which is more of a water borne disease. That means through water that is spread around. Then there is
like the tail is for moving in water, right?
Yeah. So most of these structure that are present, which are looking like hair-like projections, they help in the movement of these bacteria. And then there are these spirochetes, which are like spiral-shaped bacteria. So you can remember. So if you look at this particular Streptococcus pneumoniae, is the bacteria which causes pneumonia, right? Clostridium tetani is the one which causes tetanus, right? I know that like most of you, whenever you get injured either due to the like scratches or some iron rust, and then you go and have tetanus injection. The reason being because these bacteria, they are present everywhere and they can cause like infection in the injured area. So that's what is this Clostridium tetani bacteria. Then What is this? Legionella pneumophilia. This is one type of lung disorder, lung infection that is caused by this bacteria. But again, you need not remember this. Clostridium botulinum. This is one like, again, you might have heard of Botox treatment, which is a skin treatment which people use for kind of rejuvenating their skin or trying to look more and more younger. This is very famous in the Hollywood and Bollywood film industry, right? Because they want to stay younger even at the age of 55 or so. So that's when they do this Botox treatment. So this botulinum, Clostridium botulinum, that is the one of the bacteria from kingdom Monera, which secretes botulinum toxin. Like toxin does not mean it's like always toxic. It is one of the mechanism of defense for this bacteria for itself. So that way, this botulinum toxin is extracted and then it is injected into the skin, so that will allow more and more proliferation of the cells, like more growth of cell. So that looks like young cells. So that is what is the Botox treatment. Staphylococcus aureus, again, these are quite commonly found in the hospitals. And if at all the infection happens, especially in the what we call burn patients, those who are burned due to the fire or blasts and so on, they have high chance of infection because of this bacteria. So Salmonella typhi is the one which causes typhoid. Vibrio cholerae, I said, like it causes cholera. Helicobacter pylori, it causes pyloric infection of the stomach and so on. So there are like all these pathogenic bacteria. Pathogenic means they cause certain disease. So that is the whole of the kingdom Monera. I hope I haven't missed anything. Yeah. Any questions so far? Last point is remaining. I'll talk about that. Any questions? Anyone? Question. Yeah, so depending on the degree of the burn, the skin is always exposed, right? Skin is burnt there. Skin is our first line of defense, right? Second line of defense is all our internal linings. And third line of defense is your immune cells, whatever WBCs and so on. So if the first line of defense itself is gone, then the exposure itself is increased, right? So then the chance of having those infections, not only S. aureus, that is Staphylococcus aureus, but even the other organisms, they can also cause infections. But because in that particular setup, that bacteria is more prevalent, obviously higher chance of having those infections. Okay, so one thing that I have forgotten to mention here is Monera has been divided into two categories. Earlier times, not anymore. We don't really follow those. So there is something called as domain, right? So above the kingdom, we know that dear King Philip. So first one was the domain. So within the domain, there were different subcategories. And one of them, rather two of them, they were this Archaebacteria and Eubacteria. Archaebacteria, these are the earliest form of the bacteria, and Eubacteria, EU stands from true bacteria, right? EU. Eubacteria, yeah, Europe. So that stands for true bacteria. So Archaebacteria, these are earliest forms. That means at the quite like very long time ago, almost millions of years ago, the surface of the earth was little hotter, the temperature was higher, plus the conditions were not so proactive for letting the life survive. Not millions, but even before that. So that way, these bacteria, they were still surviving at those extremes of condition, and they are called as Archaebacteria. And one of the example of these bacteria is methanogens. These are the methane producing organisms or methane producing bacteria in short. They can also live in marshy land. They can also survive in the deep layers of the ocean where oxygen level as well as the sunlight is almost next to zero. So that way, or even in hot temperature or even in very cold temperature. So all these bacteria, they have been classified as archaebacteria, and all the others, they are classified as true bacteria, which are like subtypes of the kingdom Monera, but not from the kingdom perspective, but this was earlier used when they were studying of the domain classification. So let me just show you one diagram. So domains of life. Yeah, so for example, if you see this diagram, so I hope you are able to see it. Yeah, it's little blurred, but it's okay. So you can see earlier bacteria, archaea, and eukarya. These were the three different categories, and these were the domains of life. What is called as three domains of life, and so on. Then again later on it became separate, and from that what came is the five kingdom classification. So these were the true bacteria, these were the archaebacteria, and all of the rest of the things they were kept in the eukarya. That's when the animal, fungi, and plant, all of these were put together in one of the group of eukaryotes. But then later on, it was realized obviously this is not the case, and these all of these they possess similar categories. So that's when they were put into one kingdom called Monera. So all these organisms that you see, like you can see here, what is it? Thermococalus. Yeah. So these are the extreme temperature bearing organism, extreme halophiles. These are again living in the very deep marshy lands, or even this methanomicrobiotes. So these ones, they live in the extremes of the methane producing lands. So that way, these were the different kingdoms which were used earlier. So that's where the archaebacteria and the eubacteria, they are coming from. Yeah, EU.
But you said true bacteria.
True bacteria. Okay, so that's all about this kingdom Monera. There is a small video of 1 minute. You can watch it on your own. I will not.
We can watch it.
Yeah, I will show it to you tomorrow. Bye. So then the next kingdom that we will see is kingdom Protista. So like I said at the start of kingdom Monera, when we are classifying the organism from less complex to more complex, that was the organization. So Monera was the simplest kingdom. So the next one to Monera is kingdom Protista. So kingdom Monera was unicellular prokaryotic, and Protista is unicellular eukaryotic. So a little complex than the kingdom Monera. It was also earlier called as Protista, but then that is no more used. It's best that you remember only Protista and nothing else. So these are single-celled organisms, a well-defined nucleus because they are eukaryote with a nuclear membrane, and they includes both unicellular green autotrophic organism, or the example is Chlamydomonas, or they can also include unicellular non-green heterotrophic organism. So again, the type of nutrition that happens in Protista is autotrophic as well as heterotrophic, same like kingdom Monera. And in this, the Chlamydomonas is the autotrophic one. The Euglena, Amoeba, Paramecium, these are all heterotrophic organism, and some of their multicellular descendants like Pandorina, they are green algae. That means over the time of evolution, these green algae, they have been evolved from this category of animal, that is kingdom Protista. I assume that you would have heard of amoeba earlier for sure, even to some extent paramecium. These two would be quite new to you, that is Euglena and Chlamydomonas. So these are the different examples of kingdom Protista. Again, remember, you will have to know all these names with correct spellings. Yes, Yajnesh?
So what are those antennas?
They are the flagella.
Oh, but they're so small. Looks like antennas.
These ones, no?
Yeah.
And even these one. So these are the structures which help these organisms in movement. Like if you see it here, you can also see some small hair-like structure around the wall or the around the outermost membrane, right? So that is what we know as cilia. So these organisms, that is the next point actually. Some of them, they have cilia or flagella for mobility. So whenever they have to move from one place to the other place, they need to have these structures called cilia or flagella, and that's when it helps for them to move from one place to the other one. And sexual reproduction by a process of cell fusion or and zygote formation. So some of them they reproduce sexually, some of them they reproduce asexually. That is like sometimes they just undergo a binary fission. So they will duplicate everything, they will accumulate everything, and then they will separate into two cells. That is what is binary fission. When we talk of sexual reproduction, that means there is a male gamete, female gamete. They will fuse, they will form a zygote, and then they will grow into the next cell. So that way, there are different processes, but most of these they undergo what we call as sexual as well as asexual mode of reproduction. Again, any question here so far before we go into different subtypes of kingdom Protista? Or yeah, I guess we should do that one. Okay, so I assume there are no questions. So then there are these 5 main types of kingdom Protista or subgroups within kingdom Protista. First one is chrysophytes. Chrysophytes. This includes the golden algae or even the diatoms. Now, golden algae, I'm sure you all would have seen somewhere in the water bodies because they are aquatic in nature. What is it? Chryso? No, let me search, or no, let me show you chrysophyte first. Chrysophytes. Yeah. So these are all different examples. You will find them not commonly around us because these are deep water containing like living organisms. But let me show you golden algae. This is surely you would have seen somewhere. Golden algae chrysophytes. Something like this. In the water bodies where there are very like kind of they have been growing for long, you will find some structures like this. And if you look closely to them, they look like this. That is a very high resolution picture of this golden algae. So that's exactly how this. I'm not sure if this is algae, but yeah, quite possible. So these are the examples of golden algae. Again, I'm not sure this. This looks like some representation. Done. Yeah, these ones. Yeah. So these are all the golden algae, and obviously you know why they are called golden, because of their color, which is very obvious. So that is one example of this chrysophytes. And the diatoms. Diatoms, again, I'm not sure if you would have seen at all, but let me show it to you. Diatoms. So diatoms, they are where is the better picture of it? Okay, let me just write diatoms. Yeah, so these ones. Diatoms, they can be of any shape. Usually they are spherical. These ones are the exact diatoms. Again, these look like some leads being kept off at one place of the bottles or whatsoever, but these are actually the very microscopic structures, which are like two spherical discs, one on top of each other, and that's how they are called as diatoms, like two in a way. So two in a group, and that's what is the diatom, which is one type of chrysophytes. And again, like I said, they can be of different shapes to some extent. So that is chrysophyte. Then dinoflagellate. Dinoflagellates, these are usually photosynthetic and they are marine. That means they are growing in the aquatic bodies. They can have a different color pigments, and that's how their colors will be different. So dinoflagellate is very interesting, and I'm sure you would have heard this phenomena of somewhere this particular part, right? You might have heard that particular sea beach, it looking like a colorful beach at night, and the reason being that they have this dinoflagellate of blue color being present there, right? So, and dinoflagellate, they ideally look like this, something like this. So they can have different again shapes. So these are again the other dinoflagellate. These are all different types of dinoflagellates. They are called so because they have flagellar structures. You can see these extended projections. These are the flagella-like structures, and that's why they are dinoflagellates. And the most important why they show this luminescence, or what we call as color, is because of the phenomena of bioluminescence. So even this one, you see, this is a water body and it's looking like very colorful at night. So that's the
Can you repeat the term? Bio what? Bioluminescence.
Yeah, where was that? Here, I guess. Yeah, bioluminescence. Luminescence is a phenomenon where, like we know, a certain light is reflected by a particular molecule, and that's what is the phenomena of luminescence. That means they absorb at certain wavelength and they reflect at certain wavelength, and that reflected wavelength is what we see. So in this case, what they have reflected is the blue light, and that is what we see in terms of the color. So I'm sure you would have heard of this. What is it? Fireflies, right? Which are green in color, right? That is because they have this protein which is called as green fluorescent protein. And so these ones are the externally added into the cells, and that's why the cells will look green and so on. But again, we need not go into those complications. So there are several such proteins, what we call as green fluorescent protein. You can see this is another interesting example. Within a frog, they have injected certain protein along with this GFP, green fluorescent protein, and that's when the frog also shows the fluorescence or bioluminescence phenomena. So that's like a common experiment. These are like very common experiment done in the experimental biology setup. So then there is also something called as red fluorescent protein, which is again used in the similar way. So that is, these are the different types of fluorescent protein present around us. And that is the one which gives color to this dinoflagellate. So they can be of different color: red, blue, brown, green, yellow, and so on, right? So that is second category or second group within kingdom Protista, that is dinoflagellates. Now the next one is euglenoids. Again, euglenoids, you may not have heard of them before, but these are from a freshwater habitat, and like especially like a motionless, that means non-moving water body. The cell wall is absent, but they have a special protein rich layer which is called as pellicle, right? So I will show you the euglenoids. euglenoids. So these euglenoids, you can see here very clearly, the outermost membrane is kind of very transparent or jelly-like. That is what is protein-rich layer, and this is the whole of the euglena body. The structure is like this or like this. Yeah, you can see this is the structure of euglena. So this is a flagella-like structure, which is again helping in the movement of this what we call as protist molecule or protist organism. So that is the euglenoid category. So what you need to remember from all of this is one example of each of this category, and of course the names of this category. So that is euglenoids. So there are quite a lot to know within each category, but we are not again doing it that in detail. You just need to remember the names and example for the time. Then slime molds. So slime molds, these are specifically saprophytic. Saprophytic, like I said, they are growing on the decaying or dead organism, and the body moves along the putrefying leaves. Putrefying means again decaying leaves. So there is a difference between putrefaction and there is one more terminology. So one is organic degradation and the other one is not organic, aerobic degradation, and the other one is anaerobic degradation. So one happens in the presence of oxygen and the other happens in the absence of oxygen, and that's where the distinction is. So but again, you need not remember that. What you need to remember, putrefaction or putrefaction, this is the process of degradation of the leaves, which will ultimately convert them into completely organic matter. So that is what is the putrefaction process. So the slime molds will grow onto this putrefying leaves or the twigs or branches, and then they will nourish themselves from this organic material, and they accumulate at one particular place, and that is what is the slime mold. So again, let me show you this particular slime mold. This is also very commonly seen around you, something like this. On the leaf, you might have seen something like this growing, especially this one, on the even on the rocks or on the surface bark of the tree. You would have seen growing like this. So that is nothing but your slime mold. And if you look at microscopically, they look like this. Yeah. These are the structures of the slime molds. You can see this is brainless smile mold. Wow. But yeah, these are the slime molds. Yes. Sorry. On the rocks, some of them can be slime molds, but some of them they are this which is called as not moss. These ones. Okay. Okay. So slime mold, I assume everyone knows now. So you need to remember again the example of the slime mold. How does it look like? And then the last category within kingdom Protista is protozoa. These are heterotrophic, that means they are dependent for nutrition on others, and they can either survive as parasites, that means totally within the other organism's body, and or the predators, that means they kind of hunt. Like for example, amoeba, you might know that it engulfs whole of the organism in the sense whole of the microscopic organism for its own ingestion or digestion process. So again, no, I don't have to show this. We know this amoeba, parasium, they are here. So these are the protozoa type of organism. Any question again? Anyone? Not this. So again, there is a small video kept here. You can watch it on your own. It's not much to explain there. It's self-explanatory. and you can watch it. So next class we will start with the fungi. For now we will stop here. If anyone has any question, you can ask. Otherwise,